like that. Okay. Any question about the previous chapter? No? So, okay. So today we are going to talk about the modifications of Mendel's principles. Okay. One thing is that when Mendel did this experiment, during that time he had the concept of only certain factors, but he was very lucky. Lucky means he got some factors, those 7 factors, which were situated in different chromosomes and which were autosomal. Okay, so we knew that from the sex-linked inheritance that Mendel's actual, that is proportions, is not working there, as because it is sex-linked, not the autosomal. So he was really, it is open. So he was really lucky in that case. So after Mendel's discovery, that is a rediscovery, that when people thought that, okay, this is a very good thing in some kind of areas, particularly in the pea plants, it is working very well. So they tried everything with the Drosophila, human, and some other plants and animals, that whether this is working or not. That is the really good thing, because everybody will test that. But the thing is that it was working very well in such case. But there are some differences came out, and that was a lot. One thing is that this one, these are 2 types of snails. Okay, one is the coiling is clockwise coiling. So this is called the dextral. Another is anti-clockwise. Okay, and this is called sinistral. So what is the factor of these two, dextral and sinistral? But they are present in the population. Dextral snails are wild type in population. Sinistral are very less. Now, what are those things? It has been found that they are not following Mendelian principle, and there is something there which is not determined by only the gene or the factors what Mendel proposed. Okay. And that was the first thing. This is called the genetic maternal effect. We'll talk about this one later. So this was the first thing people discovered, that Mendelian principle is not working. Another is the sex-linked one. So what are those things? First thing is that there are 3 terms. One is called complete dominance, incomplete dominance, and codominance. Okay. So what is complete dominance? What we did before, that is, we were taking 2 plants, like it's tall and short, and we got this one, the phenotype is tall. Okay, in F1 generation. So that means there are 2 alleles for a particular character or a particular gene. There are 2 alleles. One is tall, one is short. When they are in the heterozygous condition, they are having both the alleles in the same location, but one character of this tall allele is submitting the character of sodium. Totally. Totally. There is no present. You cannot identify the difference between this heterozygous and this homozygous tall with just seeing the phenotypes. Okay. So it is totally dominating the character of the recessive allele. This is called complete dominance. Okay. But there are certain other cases which is called incomplete dominance. So that is the example of complete dominance. So when there is a red flower and white flower, if red is dominant over white, in the heterozygous condition, you will find only red flower. And in the F2 generation, when there is two recessive characters, then you will find the white flowers. That is the complete dominance. But what is incomplete dominance? That when it is in heterozygous condition, you can have any variety of color between red and white. Okay, any variety of color between red and white, and it is generally found in snapdragon flower. Okay, and the intermediate color is pink. The mixture of red and white, that is pink. Okay, this is called incomplete dominance. That means that dominant character is not expressed fully. So what is incomplete dominance? The dominant character is not expressed fully. Okay. So this is another example of that. So we have two fruits. The purple fruit, eggplant, that is the dominant character, capital P and capital P, and that is homozygous. And white eggplant, that is the recessive character. When we cross those two, we'll have the heterozygous, that is the capital P and small p. But that color is violet, a mixture of purple and white. Okay. And later, when we will find in the F2 generation, those two heterozygotes, they are violet in color. The homozygote is purple in color, and the homozygous recessive is white in color. So the condition is that here, this capital P violet color is not fully expressed. I am repeating this one a lot of times, that not fully expressed, because when I will come about the codominance, then we will find these differences. What is the fully expression and what is half expression? Okay, so this is an example of incomplete dominance, and it is not following Mendelian ratio. In case of Mendelian ratio, we should find 3:1 ratio, but here the ratio is 1:2:1. Right? Homozygous is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So it is not following Mendelian ratio. Now, what is codominance? So complete dominance means the dominant character will suppress the recessive character fully. There is no expression of recessive character in the heterozygous form. Incomplete dominance means the character between the homozygous, that is dominant and recessive, the dominant is not fully expressed. Codominance means both the allele will fully express. Okay, this is the difference between incomplete dominance and complete codominance. In codominance, both the allele will fully express, but in incomplete dominance, the dominant character is not expressed fully. So this is one thing I'll talk about the codominance. Now, in some other cases, the incomplete dominance is something like that. There are certain other factors also. Not only the genetic factors, not only like that, because this is cystic fibrosis. What is cystic fibrosis? It is a kind of disease that it is creating this gene, the cystic fibrosis gene is creating one kind of protein, and that protein is helping the chloride channel of our plasma membrane. Do you know chloride channel? So chloride channel is something like that. In the plasma membrane, there is one protein, that is the transport protein, which is called the carrier protein generally. And this is generally active transport, that is requires some kind of energy or ATP. What is the function of that? It removes the chloride ions from inside the cell to outside and taking the sodium ions inside, sodium and potassium ions inside. That is the active transport. Okay. So this CFTR protein is, if it is in the full form, that is in the wild type or normal type, they are not affecting this chloride protein anyway, this transport protein. But if it is mutant, what is happening that this transport protein is affected. In such case, it cannot pass the chloride ions from inside the cell to outside. So there is a deposition of chloride ions inside the cell. Due to that deposition of chloride ions inside the cell, there is the formation of mucus, lot of mucus. And that mucus can come to the bronchial cavity. So from the lungs, that is the alveoli, from alveoli to the bronchial cavity, and then we are coming to the trachea and that kind of things. So there will be a lot of mucus in the bronchial cavity and the tracheal passage, respiratory passage, and which is creating the respiratory disorder. Generally, cystic fibrosis people, they have these kind of disorders. They have the very big respiratory problems. Even that can be deadly. Okay, but in case, so this CF, if there are two genes, two alleles, they are both are CF. In such case, they occur the cystic fibrosis. But the question is that in case of heterozygous, what is happening? Will it be in the middle or something? Here the factor is that if it is heterozygous condition, that means plus and CF, in this condition, this protein, this gene is secreting some kind of protein, that is a genetic expression, gene expressed through the formation of proteins. The enough type of protein they could form, just a single gene, they can activate this chloride channel. Okay, so in the heterozygous, according to the, in homozygous condition, they are fine. When they have the both plus, the protein is producing in normally. There is no problem. They can activate the chloride channel, and the sodium, potassium, and chlorine, they can move through the passage. There will be no problem. No deposition of chloride ion. But if only one gene or only one allele is present, they can produce same amount of protein that can activate the chloride channel. So though it is heterozygous, but there is no such problem for the patients there. So this is another factor, not only the genetic composition, but the other factor, the formation of protein, amount of protein they can secrete, they can synthesize. That is another factor. Now there are two other things. They are called penetrance and expressivity. So what is penetrance? The percentage of individuals having a particular genotype that express the expected phenotype. Okay. So suppose there is a gene which can express certain kind of disease, but a lot of people cannot have the disease, though having the gene. That is called the penetrance. And expressivity is something like that, the degree of expression. That sometimes it can create a more, sometimes it is less. So this is the difference. Now, this is one kind of gene which is dominant in human. It is called polydactyly. Polydactyly means extra one finger or extra toe. That is called the polydactyly. Dactyly means fingers. That is the so they can have 6 fingers in either hand or toe. But the condition is that polydactyly is dominant. Okay, it is a dominant character, but people generally, if it is dominant character, so there is a very good chance that people can have this polydactyly gene in their chromosomes. That is fine. But it is found that though people have polydactyly genes in their cells, but they don't show polydactyly as a phenotype. Okay, they have the gene, they are inside the body, but they are not showing any kind of phenotypes there. Okay, so what is penetrance? They suppose another example is that BRCA mutation. BRCA mutation is BRCA1 and BRCA2. there are two genes which are responsible for the breast cancer. Okay, a lot of people have this BRCA mutation. A lot of ladies they can have this BRCA mutation. Even the breast cancer is also in males also, because there are only two phenotypes we have found, and that is in Cincinnati, two male breast cancer patients. Okay, so they have the breast cancer gene, may be mutated, but they don't have breast cancer. Okay, so the thing is that in such a condition, they have the polydactyly gene, but they are not polydactylous. Okay, so now we need to calculate that how much. If 42 people having that allele and 31 expressed the polydactyly, then the penetrance is about 90%. So that means if you have the gene, you might not have the disease also. So we need to think about the penetrance there. This is called penetrance. Genotypically, they have the gene, but there is no expression in phenotypes. So how much expression is there of that gene phenotypically, that is called the penetrance. Clear? Now, what is expressivity? That is, this is the example. The brown animals, the genotype is BB. And brown means there are various sets of browns. Okay? So in which set they will express? And you can find this one very useful here. So this is the example of penetrance and expressivity. Penetrance means either they have the purple or white. There are two options. So 1, 2, 3, 4, 5, 6, 7, 8, 9. Among 9, 1, 2, 3, 4, they are not showing any kind of activity there. Okay. 5 are purple, 4 are white. Only two of things, but all of them have the same gene. Either they expressed or not expressed. What is the expressivity? They have various types of sets here. They are purple, okay, that's fine, but various sets are there. That is the expressivity. So they can express, but in different sets. So when there are both conditions are there, they have either different sets or no set at all. That is the variable penetrance and expressivity. Can I give some example of that one? Penetrance and expressivity. Expressivity. Generally the skin color of human. They have the same gene, but that is multifactorial also. But they have these kind of variations, particularly for that. And this pattern. This is variable expressivity. That is this color and this black patch, the patches, they are variable in different patterns. Okay, both of all these dogs, they have the same gene there, but their expression is different in different patterns. And this is very complex system. People didn't know about that, but they are the same things, but why they have different patches. And then the concept of expressivity came. Now think about that. Assume that long fingers are inherited as a recessive trait with 80% penetrance. Previously we thought that okay, it is expressed, but right now you have another condition, it is 80% penetrance. Two people heterozygous for long fingers mate. What is the probability that their first child will have long fingers? What do you think? Any idea? First thing is that they are heterozygous and long fingers is a recessive trait. Okay. So for long finger, we take small l, and that is recessive. For normal finger, it is L and which is dominant. They are heterozygous for the trait. So LL and they are the heterozygous for the trait. I can write this way. Okay. Now they are crossed. If this is the thing, we can have the 4 different patterns here from female. Oof. Okay, so we can have LL like this. So this one is long finger. This is it, right? This one is long finger, all other normals. So what is the proportion of getting long finger if the cross is normal? 25%. Here, this is 25% or 1/4. 1/4. Now the penetrance is 80%. So 80% of this 1/4 can show the trait. Clear right now? They have the allele. 1/4 of this population can have the allele, but only 80% of them can show the trait. Another extra thing here. So 1/4 into 0.8. That should be our percentage. They can show the trait. That is only the 20%. Okay. So the allele, we can have the allele in 25% of the people, but among 25% of the people, only 80% can show the trait. So as a whole, totally 20% people can show the trait. I won't do the too much problem in this because it is itself problematic. There are a lot of things. Okay. So that is the penetrance, expressivity. I didn't talk about the codominance yet. Incomplete dominance and complete dominance. The 4 things. Now it is called the lethal allele. Lethal means death. Okay. Death allele. What is that? This is called the yellow mouse. Okay. Yellow color, yellow skin color mouse. And they are in the heterozygous conditions, found in the population as in the heterozygous condition. Why? When these 2 heterozygous mouse is crossed, we can find the heterozygous and non-yellow mouse. But in the population, we cannot find any homozygous yellow mouse. Okay. When they are in the homozygous condition, those yellow genes, they are lethal. They cannot survive in the population. Okay. So there is no such ratio of 3:1, but we are getting only 2/3 of progeny are yellow and 1/3 progeny is non-yellow. There is no homozygous yellow mice here. This is called the lethality or lethal allele. So when they are in the homozygous condition, they are lethal. They are not expressing in the population. Now, a cross between 2 green corn plants is 2/3 progeny that are green and 1/3 progeny that are white. What is the genotype of the green progeny and the white? If I think the white is the dominant character. answer? Yes.
The green would be heterozygous, and the yellow would be homozygous recessive.
White.
Wait, yeah, white would be homozygous recessive.
No, actually, it is white is homozygous dominant, but you are very close. You are very close, just opposite. You took the homozygous and recessive, but you are very close. So that is white genotype is dominant here. The corn is white generally, and the green genotype is heterozygous, but the homozygous recessive is lethal. Yeah, that's why we got one-third population. Okay, that is the thing. That is the lethality. That is lethal. That's why we got one-third population rather than having one-fourth. Okay, now is the multiple alleler codominance. Now, in case of incomplete dominance, we have found that if it is red and white, it is a mixture between that two. Okay, so red is not fully expressed, and that's why it is pink. But here in codominance, there are two different alleles in the same loci, but they are expressing fully, both of them expressing fully. And that is the example of AB blood group. Okay, so in such case, what we are getting here, we have four blood types. So one is A, B, AB, and O. There are four blood types. Alleles for these blood types for A is generally we write it down as IA. Okay, that is the allele for A blood type. For B it is IB, and for O it is recessive small I. Okay, these are the allelic composition. Now, if the blood type is A, it could be, the genotype could be IA, or IA and small I. Okay, either it is heterozygous condition with the recessive allele, or it is dominant homozygous. Okay, in case of B, that is the same thing again, IB, or IB small I. Okay. For the O, it is completely recessive, so homozygous recessive. Now the thing about the AB one. Okay. For the AB, they have the composition like this. IA and IB. Both IA and IB are creating their own characteristics in the blood. That's why the blood group is AB. And what are those? They are producing here. They have no antigen here, particularly for the O blood groups. So they can give it to any other person. And AB blood group, they have no antibodies. What is the function of that? Antibody is protecting us from any kind of antigen. Okay, so antibody is fighting with any kind of antigen. When there is AB, suppose A blood group, they have the anti-B. That is anti-B antibody, that is antigen A. A is for antigen A, and antibody beta. Generally, it is called beta. Alpha, beta, those are the antibodies. So they have the antibody beta. Now, antibody beta can prevent the entry of antigen B. Clear? Antibody beta can prevent the entry of antigen B. So that means if antigen B is given to this person who has the antigen A and antibody beta, the blood will coagulate. Okay? So A person, blood group A, cannot receive the blood from B. Okay? B blood group cannot receive the blood from A because it is having antigen alpha. which can prevent the entry of antigen A. Now, for this one, they have no antibodies at all. This group, AB group, they have no antibodies. If there is no antibodies, they cannot block any kind of antigen. So any kind of antigen they can receive. Okay, so they can receive any kind of antigen. AB blood group, they can receive any kind of antigen. This is called universal receiver. O blood group, they have only antibodies, but they have antibodies but no antigen. If they have no antigen, suppose O blood group, if I will give O blood group to A, they have no antigen at all. A blood group having antibody beta, but beta will not fight with any kind of antigen because there is no antigen at all, so they can receive that blood. Okay, so O blood group is called universal donor. They can give blood to any person and there will be no problem. AB can receive blood from any person, there will be no problem. So this is AB blood group is called universal receiver because they don't have any kind of antibodies. O blood group is universal donor. They have no antigen at all. Okay. So right now, based on this condition, this A and B both are expressed here. That's why these are called codominants. Now clear the incomplete dominance versus codominance? Any confusion between these two? There is another blood group. It was previously thought that during 1970s, there is another group called the MN blood group. Generally, it is not very used right now. In MN blood group, they are also codominant. Okay? There are two things. One is M, another is N. When both are together, M and N, the blood group will be MN. Okay? This is also an example of codominance. This is another example. Now, what blood types are possible among the children of a cross between a man who is blood type A and a woman blood type B? Man, blood type A, woman, blood type B. Any answer? Could be any of them, yeah, that is right. But in which condition?
They were like heterozygous.
Okay, if they are heterozygous. So if they are homozygous, at first I need to think about that one. If they are homozygous, so that is IA and IA here, and this is IB and IB. Okay, in such case, there is only one probability having AB blood type. Because one IA from here, one IB from here, so the blood type will be AB. Okay, this is one possibility. Now, if this is heterozygous, this one is only heterozygous, this one is homozygous. In such case, or either case, B is heterozygous and A is homozygous, any of these cases, what will happen? We can have two patterns here. So from male, we'll have two types of genes, two types of alleles here, and from female, we have only one. So this is AB, and this one is EON. Okay, that is another possibility. If the female is heterozygous and male is homozygous, in such case, we'll find only A here and AB here, just opposite. Okay, if one is heterozygous. If both are heterozygous, then what will happen?
We'll have one A, one B, one A, and then one O.
All the blood groups are possible. All the blood groups. This one is AB, both are codominant. This one is B, this one is A, and this one is O. So all the blood groups are possible. What about this one? There are 4 children having A, B, O, and AB blood type, and there are 4 sets of parents here. Whose child is which? Let me find out. Write O here, then take O here. If you use what is this formation, then use not that formation. Don't mix these two up. Any answer? This is like private investigator. You have to find out. I have found 4 children here. I found 4 sets of parents. I need to give them the children. Waits for what? Detective. Yeah, really, actually, for the forensic laboratory, generally they are testing these things. Who is the father of this child? In such kind of cases, they use the blood group. And not only that, right now DNA fingerprinting. That is another part. So let's go for this one. Okay. First of all, I am going to the D. A, B, C, D. Four problems are there. Go to the D. O versus O. O versus O means all are recessive. Okay. So there is no way the dominant character will come. So this should be O. That is gone. Right now we have A, B, and AB. Okay. The second one is here A versus AB. In such case, what are the possibilities? If it is recessive, that means AO condition, and this is AB condition. So we can have one A, one B, and one AB. So like A, O, and from here we can find these two. So all four times, that is possible. Okay. From this cross A and 0. If it is homozygous, we can find only A. That is this versus this. We can find only A. If this is heterozygous, then we can find one A0, another OO. Okay, either A or O. So the question is that in both the cases we can get A. So these children should be A because already we got O. So this child should be A anyway. For the first one, that is AB and O. So AB and O means either we'll get AO or BO. But there is no way we can get AB. Okay. If I will not get any AB here, in both cases, these two parents are confusing because we can get B blood group in both cases, and already we got the A1, so there is no question right now. But we got B blood group in both cases. But who is the father of AB blood group? Only one person is there. Only one set is here who would be the father of AB blood group. So this person must have the AB child. There is no other way. There is no other possibility. Other parents cannot be the AB, father or mother of AB child. So these parents should be the parents of AB child. Then only the B is left. So we can take the B from here. This should be the explanation. So we have a lot of possibilities, but you have to rule out all the possibilities to make a perfect choice. At first, I ruled out the possibility of O because O is O. That's fine. Then there are a lot of chances, but we need to figure out in this way. Okay. I like this problem. You can use so many brain activities there. Okay. Now, another thing is called gene interaction. Okay. Gene interaction takes place when genes on multiple loci determine a single phenotype. This is a little bit more critical. So gene had multiple loci, but they are determining a single phenotype. So what are these? There are two things. One is gene interaction produces novel phenotypes, and gene interaction with epistasis. Epistasis is a very common known thing here. So this is one example, gene interaction. In the capsicum, capsicum is the scientific name of pepper. So in capsicum, we can find red, peach, orange, and white type. All these four types are present in the market. Okay. So for the red gene, this is dihybrid cross. So there are two genes here. As a full dominant is Y plus YY or CC. This is a full dominant. And full recessive is YY CC. This is white and this is red. Okay. When we got the heterozygous, the red color is present in case of heterozygous condition. But so when the red color is present, if both dominant genes are present, then the color will be red. So we figured it out that Y plus and C plus, with whatever gene it is, that doesn't matter. If both dominant genes are present here, the color will be red. If Y plus, that is one yellow Y gene is present, and these two recessive genes, two CC recessive genes are present, then the color will be peach. So these two recessive genes are determining the peach color. When these two recessive genes are there, they are determining the orange color with this dominant one. Only one dominant, that is fine. And when both are recessive, that is white. So that is the variation of their genetic composition. They can determine the different colors. This is called a gene interaction. Dose of gene. When both are recessive, they are determining a different patterns with only one dominant gene. When both are recessive, they are determining one color pattern when all of them are recessive. Okay? And when only both of them, only one set of dominant here, they are determining one kind of colors. This is called gene interaction. Definitely it is not following Mendelian machine. Okay? And what is epistasis? Epistasis is a little bit more different, that there are two genes in two different areas. So suppose one gene is here, one gene is here. This gene is producing some kind of protein, that is protein 1, but this gene is determining whether this protein will be present or not. Okay? So this interaction is something like that. One gene is masking the effect of another gene. Okay? This is an example that H factor in our blood group, human blood group, H factor. H factor, the function of H factor is that producing one kind of H protein here, which is attached to this one. If the H protein is present, then either A or B can attach to that one. If H protein is not present, then there is no way A or B antigen can attach to this one, this protein. So H protein is determining only one thing, whether A or B will be attached or not. Okay. Suppose I have A and B, but they cannot be attached because that H protein is not here. So H protein is actually determining that factor whether this A and B antigen will be attached here. It is masking the effect of that one. So we cannot just determine having the A or B here. Okay. Another big example, this is from your book, but this is from other book. That's why I put it as a slide here. Coat color in Labrador. Okay. P is for the black pigmentation. Small b is for the brown pigmentation. Okay. E is for the deposition of the pigment. Whether the pigment will be deposited or not. They can produce the black color, but whether they will be deposited or not. And E, no pigment deposited. So if capital is E here, suppose E is a black color, when the capital E is here, it will determine the black color will be deposited. If there is small e here, then it will determine that there will be no deposition of black color. So if there is no deposition of this color, that means the color will be white. Clear this part? This is actually Present. This is present. The black and brown color is present there. There is no problem. Those genes are there. But one gene is determining whether it will be, I can coat it or I cannot coat it. I have the color in my hand. I have the color in my hand, but that E gene is telling me whether you can coat it or not coat it. So the factor is something like that. Think like in that way. Okay. I have two colors in my hand. I have a paint brush. Then either I can coat with that color or I cannot coat it. So in such case, when there are small e in the recessive condition, all these cases are white because they will not allow to deposit the pigmentation. Whether it is small b, whether it is capital B, that doesn't matter. If it is small b, that means it will be brown. If it is capital B, that means it should be black. But there is no deposition of the color. That's why it is white. So the total percentage is different. Rather than having 9:3:3:1, we are getting 9:3:4 of this gene only. So this is masking the function of brown and black gene. This is the great example of epistasis. Okay. So to understand this one, if you can remember these things, you can understand the recessive epistasis very well. And this is in the recessive condition. That's why it is called recessive epistasis. Okay. Clear? And dominant epistasis. Dominant epistasis means when they are in the dominant condition, they will produce white squash. Any of the gene is present there, they will produce white squash. What is their function? The function is that when this dominant allele is there, they cannot convert the color. There are biochemical pathways. So compound A, they need one enzyme to convert that color into green color. That is called the compound B. This is the pathway. Okay. Compound B need another enzyme to convert the color to compound C. That is the yellow color. Now, if this enzyme is not present, what will happen? There will be no conversion of green color or yellow color. Okay, if this enzyme is not present. If this enzyme is not present, we can find that white color and green color, but there will be no yellow color. Okay. These genes, that is enzymes are encoded by genes. They are proteins, they are encoded by genes. And W genes, that is one dominant W, they can inhibit the formation of enzyme 1. So if W is present, white squash, because they cannot convert discoloration. They will totally block the whole pathway. They will inhibit the formation of enzyme. If there is no enzyme 1, there is no change in coloration to green or yellow. Okay. So when the W is present, they will produce white squash. Whatever the gene is there, that doesn't matter. So this gene is dominant. That's why it is called dominant epistasis. Clear? This is another thing, the duplicate recessive epistasis, the same formation, but it is in the both recessive condition. I just put it there. So there are the things. So in the Mendelian ratio, we know the normal ratio for any kind of dihybrid cross is 9:3:3:1. But there are different patterns here. When there is recessive epistasis, the cross is that is proportion is 9:3:4. If it is dominant epistasis, we have 12:3:1. If it's duplicate recessive, then 9:7. Duplicate interaction 9:6:1, and dominant and recessive 13:3. So these are the all patterns when the Mendelian ratio is not properly followed. And these are the, so that means these are all autosomal. One thing is there. Another is called complementation. So complementation means that is whether the mutation are at the same locus or different locus. And this is a little bit complex. I'm just giving you the examples of complementation. Agouti, black, extension, and spotting. This is actually tri-hybrid cross. So coat color is determined the number of genes. One is A and SS, but adding only B and E loci. Okay, so most bagels have this kind of genotypes. Carnassians have this kind of genotypes adding at B loci. Just know these things because this is a lot of complexity here. Okay, and this is called the complementation in case of three, even four genes are responsible. Now there are two main patterns. One is called sex-influenced factor, and one is called the sex-limited factor. Sex-influenced, the term means one character is influenced by a particular sex. Sex-limited, that means that character is present only in one sex, not in both sexes. Okay, there are two things here. The sex-influenced state, it is the beard of goat. Okay, so when their genotype is P plus B plus, that is beardless. That is the normal dominant characteristics. The goats are beardless. In case of males, in case of females, both are same. Okay, when they have in the heterozygous condition, then males are bearded, but females are beardless, having the same heterozygous condition, but they are having two different characteristics here. When they are in recessive condition, both are bearded, so there is no problem at all. Problem is there when there is heterozygous condition, only male is affected with the beard, but the female is not affected. So that character, the beard characters, is influenced by the sex male here. Okay, so this is called sex influenced trait. This is the picture of that. And next, sex limited trait. So sex influenced trait means that character is present in both sexes, but in heterozygous condition only it is present in males. Sex limited means that character is present only in one sex, not in both sexes. Beard is present in male and female both, but in heterozygous condition, the beard is present only in male, not in female. That is sex influenced. Sex limited means this kind of male kind of pattern, tail pattern, it is not present in the females anytime. Only it is present in males. Okay, you cannot find in any kind of females. So what are the genotypes here? If it is capital H and capital H, hen feathering in both cases. So capital H is for hen feathering, hen type of feathering like this, and it is the dominant character. Okay, so when it is in heterozygous condition, if that is the dominant character, that will be the hen feathering again. Okay, because that is dominant character, so it will be expressed anyway. When it is in the recessive character, so there will be no hen feathering at all. Recessive character is only expressed in male in case of cock feathering. That is this kind of feather. Only present in males. But the same character, when the females are having the same recessive characters, they are showing the hen feathering. There is no expression of cock feathering. This is sex limited. So clear, these two? It is a little bit confusing. Sex influenced versus sex limited. Sex influenced means both have the same character, but one sex is influenced by that character in heterozygous condition. Sex limited means that character is not present in other sex, only present in the one sex. And we have in human, there is also another sex limited trait that is called the precocious puberty. Precocious puberty is something like that the male can get puberty at the age of 4. Okay, they can get the age of 4, and that is not present in female anyway. So if the precocious puberty male is crossed with normal female, then in next generation we could find there are female with heterozygous condition, but they are not showing any kind of precocious puberty. Only the male can show that precocious puberty. So this is sex limited, present only in one sex, having both same case of genotypes. Okay. Now another pattern that is deviant from the Mendelian cross. Actually, right now when we are going for the genetic engineering, gene manipulation, we are getting a lot of more things. This cytoplasmic inheritance, actually mitochondrial inheritance, we didn't know about that long before. Like during 1980s, we didn't know about MTD. Really, not too much. We knew that mtDNA is there, chloroplast DNA is there, but not the pattern of their inheritance. That mtDNA is only present generally. This inheritance is through mother. Okay, they can determine, the mother can determine that what kind of mtDNA will be there. So during this proportion, their cytoplasm, actually, this mitochondria is in cytoplasm. They can determine that what kind of pattern, what kind of mitochondrial DNA it would be. And my mother's cytoplasm is determining that pattern. Okay, that's why it is called the maternal inheritance. And when we have to trace about the human evolution, that is from where we evolved, we traced this mitochondrial DNA, and it went to only one woman in East Africa, which is called the mitochondrial ape. We all originated from that ape, all these 7 billion population in the world. Okay, and that is because it is from mother only, so it is very restricted to the mother's pattern. So it is both present in males and females. Generally, it is both present in males and females, and there are a lot of variations here because it is cytoplasmic. Each and every cell can be variant. Each and every cell can be variant. Whether what type of genotype we have, that doesn't matter. Okay. Usually inherited from one parent, generally from mother. Generally from mother, the MTP. And reciprocal process give different results, exhibit extensive phenotypic variation, even within a single family, even within a single body. It can give that kind of, because each and every cell is different. Each and every cell is different, their mitochondria is different, so it can give that kind of patterns. If it is in the zygotic way only and then stop, then it is fine. If it is continuing after that, each and every cell division can give different kind of results. And this is one example, just you can go through that one. Now there are a couple of more things. Genetic maternal effect, genomic imprinting, and epigenetics. What is genomic imprinting? Differential expression of genetic material depending on whether it is inherited from male or female. Same gene there, but if it is from male, there will be one expression. If it is from female, that will be another expression. That is called the genomic imprinting. And epigenetics is phenomena due to alterations to DNA that do not include changes in the base sequence often affected the way. Actually, we will not do the epigenetics here. So maternal effect. Maternal effect is that suppose here the first time we have one dextral snail, one sinistral cell. Okay, dextral cell is dominant, so it is having the plus characteristics. Homozygous dominant. The sinistral cell, it is recessive. There is no plus sign. Okay. When they mate, they will form the zygote, that is the heterozygous. So if it is heterozygous, the S plus character should be expressed there. So it should be dextral, right? Think about that. If the plus character is dextral in heterozygous condition, it should be dextral, right? Right or wrong? It should be dextral, but it is giving the sinistral characteristics. It is giving the sinistral characteristics. Why? Because these sinistral characteristics is from mother. As the mother is sinistral, that's why it is sinistral. Though it is heterozygous, it should be dextral, but as the mother is sinistral, then it is also sinistral. Okay. Now from there, if they self-fertilize, because snails are hermaphrodite, they have their male and female organ in the same body, they can self-fertilize by themselves. Only one animal can reproduce there. They don't need two animals. Okay. So they can produce this type of characteristics here. Okay. Now the mother has the gene S plus here. As the mother has the gene S plus, so they are dextral. And that is due to actually what is happening here. They are producing certain kind of protein in the cytoplasm. That protein is affecting their formation of this square. Okay. And that is not through gene, through cytoplasm. That's why it is called cytoplasmic inheritance. And what is genomic imprinting? Both we have this gene here, that is IGF2 and IGF2 both in male and female. Okay. And what is the function of this IGF2? That is the formation of a particular embryo. Okay. The size of the fetus is determined by this allele. And the case is that when the parental, that is from the father, this IGF gene is active and this gene is not active. This is called silence. Only this gene, the gene, same IGF2 gene from father can determine the size of the fetus. And this gene from mother is not doing anything at all. This is called gene silencing. Later we will describe this one. This is called gene silencing. One gene is silenced. They don't have any kind of effect. They cannot express at all. Okay. Another terminology is called anticipation. What is anticipation? Anticipation is that some genes are present right now in later generation, but they are not really very active right now. But previously that gene was very active. Okay. So one example is that right now the patients with moderate to severe myotonic dystrophy, they have the moderate to severe myotonic dystrophy, which is autosomal muscle disorder. They have the ancestors who were mildly affected. So they had the same gene before, in several generations before, but that gene was working not too much during that time. That was mild, they were mildly affected. But the same gene, after a couple of generations, they are very active. We really don't know what is the cause of this, but we just know this one. and a lot of research is going on. What is the formation of protein and what kind of protein is forming there and what kind of other proteins they are affecting this kind of thing. And next is temperature sensitive. Temperature is affecting the character of some organisms. Here, Here is one, that is a vestigial wing. Vestigial wing means very short wing, very small wing. This fly, that one, that is the normal wing. Okay? And this fly has vestigial wing, and this is Drosophila. So what is happening here? When the Drosophila eggs are reared below 29 degrees centigrade or 29 degrees Celsius, the flies of homozygous condition have vestigial wings, just for the temperature. When they are above 31 degrees Celsius, they are having normal wings. Okay? Gene is same, every genotype is same. There is no problem for the genes and genotypes. Only the temperature is determining whether they will have the vestigial wing or the normal. That is called the environmental effect. And another great example is that of this environmental effect, this rabbit. Himalayan allele of rabbit, they can produce this kind of coloration. The black, that is ears, nose black, and the legs are black. Okay. But the thing is that when they are reared at 25 degrees Celsius or lower, then the black color develops. But if they are reared above that, above 30 degrees Celsius, there is no black color. They have the same gene, same thing. From the same embryo, you can divide those two. You can rear one embryo in the minus 25 degrees Celsius, another in 30 degrees Celsius, and you'll find two different colors. What could be the possible explanation here? Possible explanation is that some kind of protein which is producing through these genes, they are active by that kind of temperature or not. Okay. So there are a couple of other things. Temperature sensitive mutation, a conditional mutation that produces the mutant phenotype in one temperature range and the wild phenotype in another temperature range. Like this, below 25 or above 30. Permissive temperature, the temperature at which a temperature sensitive mutant allele is expressed the same as the wild type allele. If the wild type allele is black coloration, then this 25 degrees Celsius is the permissive temperature. Restrictive temperature, the temperature at which a temperature sensitive mutation expresses the mutant phenotype. If the mutant is white, then 30 degrees is black. Or if it is vice versa, then it is something like that. This is called temperature sensitive mutation. Okay. Now there are some other characters here. I think, should I stop today? It's a lot. I know that. It's a lot. So I'm not giving you any problem with the epistasis and other things, but I will suggest that if you are interested, you can go for some problems with epistasis. So you can have the better idea of that one. There are a lot of things here. In this chapter, there are a lot of things. Just go to each and every terminology. What is sex influenced trait? What is sex limited trait? What is epistasis, recessive, dominant? Each and every terminology and try to memorize those things. And these are very little part is left here. That is the polygenic inheritance, pleiotropy. Those are very important things also. I'll discuss in the next class at the first time. Okay. And then we will go for the pedigree analysis in the next class. So we'll do a little bit problem. I like pedigree analysis very much because it is really very interesting. I think it is very interesting. What is it? Yes.
Homework or anything over the weekend?
I will